Welcome to a demonstration of the Clinical Sequence Analyzer from Nextcode Health. Click on Studies and the study will open to Study Settings. The detailed phenotype description under Description. In this case, we are analyzing the case of two sisters affected with blindness and deafness and bulbar weakness, bringing to mind Brown-Violetto-Van Leer syndrome. Under Study Settings, you can continue to add to the description, the phenotype signs and symptoms, as well as genes to add candidate genes. In the Report Settings tab, you specify the analysis parameters of your study. Specify the maximum allele frequency allowed here. Select the variant effect predictor consequences you are interested in. Under Advanced, you can manipulate the variant quality settings. ACGM category settings allow you to select pathogenic only or pathogenic and unknown variants. Genomic filters allow you to choose exonic only variants, exclude known repeat regions, or provide an exclusion variant file or exclusion region file. Under penetrance, you can choose to allow one case lacking your variant and a control possessing it, if your study has controls, that is. Under gene lists, you can choose from a number of predefined gene lists, including cancer, cardiomyopathy, and childhood recessive. You can click on any list name for gene details. Now let's begin the analysis. To begin interacting with the analysis tools, go to Candidate Genes. This view will show you all variants within genes in your candidate gene list. In this case, 8 variants, including the SLC52A2 riboflavin transporter that we have confirmed as a causative mutation in this case, are available. You can choose to view the results by genes and choose between 30 columns to populate the view. The Gene Candidate Phenotypes column tells you how the candidate is part of your analysis. Each phenotype sign and symptom, as well as each added candidate gene, represent one box filled in green, and if the box is filled in yellow, it is a paralog of a gene from the candidate gene list. You can choose to view by variants. The variant view lists genomic position, gene name, position within coding region, position in the protein, ACGM category, and the nature of the variant effect predictor consequences. You see the variant effect predictor impact, OMIM IDs, and diseases that are known to be caused by the gene. In the variant view, you can sort by any of 90 attributes. You can also go into the table view and go back to the list view. The gene coverage dialog is at an amplified scale of 50% and depicts the percentage coverage of the gene at a number of depths. Green means greater than 10, yellow is between 5 and 10, and red is less than 5. You can text search your results and open the study or the particular query in the Sequence Miner Advanced Analysis module. You can choose to bestow a star on a variant. You can choose to make a comment on a variant where you will be asked to denote clinical significance, likely mode of inheritance, and write a comment in the comment menu. You can look at all available attributes of the variant in the vertical view. For example, get the mother's call and sequence depth at that position, which would be interesting in a de novo case. At this point, you will choose what kind of inheritance model you think is likely for this study. Autosomal recessive, homozygous, and compound heterozygote will analyze all variants following an autosomal recessive inheritance pattern, including homozygous variants and compound heterozygotes. The homozygous, autosomal recessive, and compound heterozygote tabs distinctly analyze homozygous and compound variants, respectively. Dominant, X-linked recessive, and de novo candidates are self-explanatory, but missing known variants display variants missing from genes in your candidate gene lists, while all missing will report missing variants for all known genes. We believe our case to be inherited in an autosomal recessive fashion, so we select autosomal recessive, homozygous, and compound heterozygous. And the first result is this SLC52A2 riboflavin transporter gene that we have confirmed is the actual causative variant in this case. Variant or gene links open a sequence miner and a genome browser, or sync with an open sequence miner genome browser, and will allow you to eyeball the variant or entire gene in the genome browser. As you can see, the index case and her sister are homozygous, and the mother and father are heterozygous. But if the candidate genes analysis is unproductive, the next destination is the Known Variants tab, where you will see all published disease variants, and you can select variants that have been commented on by anyone in your institution. Homozygous variants that have a published disease and homozygous variants with comments in your institution. Finally, you can include all variants irrespective of their previous known connection to disease. Even here, without the use of detailed phenotypes to create candidate gene lists, though we had 509 variants to work with, sorting by ACGM category brings our known causative variant to the top of the list. Ignoring phenotype information is not recommended. As mentioned before, detailed and exhaustive phenotypes are essential in order to increase the likelihood of solving the case. The summary report is a useful feature of the Clinical Sequence Analyzer. The summary report will serve as a permanent record of the case study, as well as a medical handout and a part of the patient's medical dossier. The Overview tab has study name, description, participants, and requesting physician. Starred and commented variants reports clinical attributes on variants deemed worthy of being starred or commented on by you or anyone else working on the study. The Findings tab allows you to choose between transcripts, and the one you check will be included in the report. In the Gene Coverage tab, you see a graphical representation of what the percentage is of genes having 85, 90, and 95% of genes covered at 20x and 10x for your candidate gene list and all genes. Statistics and QC metrics shows counts and distributions for variants in the candidate gene list and in all variants. The In Distribution column tells you where in the distribution curve the variant lies when compared to all of the samples that have been processed. Click on Generate Report and a printable report is generated. Please look for additional training videos at nextcode.com.